Hello friends, today we will discuss the gram stain procedure. In my previous lecture, I already discussed the gram positive and gram negative bacterial cell wall and their composition. So today we again draw the picture gram positive and this one is gram negative bacterial cell wall. So before the going step of gram stain, this one is very useful and you must have to understand the cell wall structure. So at first when we'll start the gram stain, first is we have to make prepare a smear. I will showing with practically how we will prepare a smear. So at first we have to prepare a smear, then we put crystal violet solution over the smear area and it allowed to hold 1 minute. So now comes to the picture. So when we'll put the crystal violet area, the gram positive bacterial cell wall which mainly prepared by the peptidoglycan layer, and here this one is phospholipid glycolipid, so many layers are present. The peptidoglycan layer is present inside. Okay, so this one is lipoprotein, phospholipid, lipopolysaccharide, all are present outside, but peptidoglycan layer is present inside in case of gram negative bacteria. So when we put the crystal violet solution for gram staining procedure, and this is also called the primary staining solution. So that time gram positive bacteria will take part, it will take primary stain, the gram positive bacteria, and that time also the gram negative bacteria also take part the upper portion of the crystal violet solution. Okay, like that. Then comes to the procedure. It then wash with distilled water or tap water. Then we put the grams iodine over the smear for 1 minute. That time what happened? That time what is the function of grams of iodine? It acts as a mordant. So what is the function of mordant? It hold up the primary stain. It takes strength, the primary strength. Okay. So now here the grams iodine also mixed with the crystal violet. Here also same like that. But thing is that crystal violet and grams iodine both are combined together. Okay. After that we wash with distilled water or tap water. These are second step. What is the third step? Third step is the decolorization. It is very important step. We have to decolorize with acetone alcohol mixture. Acetone alcohol mixture. Okay. When we put the decolorizer drop by drop over the smear, that time in case of gram positive bacteria, we seen the grams iodine, it removes very less an amount, and the primary stain crystal violet, it already takes over the peptidoglycan layer in case of gram positive bacteria. That time it never be washed off. Okay, and in case of gram negative bacteria, these all are component made by the peptidoglycan, not peptidoglycan layer, phospholipid, lipoprotein, lipid molecule. So lipid can be dissolved by the alcohol mixture. So what will happen when we'll put the decolorizer? That time the primary stain with grams iodine, it removes quickly in gram negative bacteria. After that, now comes to the procedure. Again we wash it properly with distilled water or tap water. Next part, the last part is we have to put saffron in for 30 seconds. That's time if we put the saffron in case of gram positive bacteria, gram positive bacteria already takes the primary color, crystal violet color. It never takes no color, so it will remain the primary color, crystal violet color. But in case of gram negative bacteria, all color will be removed at the time of decolorization. So the peptidoglycan layer of the gram negative bacteria, it remain unstained in condition. So what will happen when we put the saffron in for 30 seconds? Why we select the 30 seconds? Because this peptidoglycan layer is too much thin. So when we will put, that time it takes the saffron color. Okay, the saffron color, this is also called the counter stain. After that we wash it properly and allow to dry. This will take part, allow to dry. So after drying, what we will observe? We observe very clearly the gram positive bacteria will takes violet in color and gram negative bacteria takes pink purple in color. We already observed in our smear. After microscopic observation, we can identify it is cocky or bacilli like this. So next, with this next video, with this gram stain procedure, I already showing the practical how it prepare. Okay, now let's come to the gram stain procedure. We are performing gram stain. Before gram stain procedure, we have to prepare a smear. Before smear preparation, we need glass slide, micrometer loop, distilled water, and obviously need culture colony. First, we have to sterilize the micrometer loop. Then allow to cool it. Then take a distilled water, a loop of distilled water. Again, we have to sterilize, red heat sterilization process. After sterilization, we have to take a little bit of smear and mix with the distilled water present in slide like this. Again, we will prepare a slide. After getting sterilization, we have to touch the media. When we touch the media, if no sound is arise from there, so we can confirm that the micro airloop is cool. Take a little bit of colonic and prepare a slide. After preparation of slide, we have to again sterilize the nichrome loop and it allow to dry with air, or we can gently heat fix like this. So these two procedure: at first air dry, the smear, after that heat fixed. Okay, what is the reason for his heat fixed? So there are two reasons. One, it can decontaminate it. Number two, at the time of washing in gram stain procedure, the bacterial cell wall easily attached with the glass slide, so it never wash off from the slide. This is prepared smear. Now we will perform gram stain. We are performing gram stain. So first step is we have to cover whole the smear area with crystal violet. After crystal violet solution, we have to hold it up to 1 minute. After completion of 1 minute, we have to discard the crystal violet solution and wash with tap water. And we should remember that tap water should not directly fall on over the smear. It should be like that. The water comes to the hands to slide smear. After primary staining with crystal violet, we have to put grams iodine and we cover the only smear area, and it also hold up to 1 minute. After 1 minute completion with grams iodine, we discard it and washed properly with tap water, same like before. After that, we have to decolorize with acetone alcohol mixer like that by one drop by drop. Now we will put the saffronin over the smear area and allow it for 30 to 40 seconds. After completion of counter stain, we have to discard again and wash it properly with tap water like before. And after that, now gram stain is ready. Now it allow to air dry. After preparation of gram stain, so we can easily differentiate between gram positive and gram negative bacteria. Here we look, the gram positive bacteria, it takes crystal violet color, and gram negative bacteria takes the saffron in color, like pink in color. So friends, we are easily differentiate gram positive, which one is gram positive and which one is gram negative bacteria. Now let's go for the microscopy. Now we are going for the microscopy.